morning or afternoon, dear ladies and gentlemen. My name is Jan Markus. I am a senior scientist and production manager at MatTek In Vitro Life Science Laboratories. And I'll be talking today, as announced, about the reconstructed 3D tissue model, which was developed by us of the small intestine, which is or might be a useful tool for assessing safety and efficacy of food products and also for other fields. First of all, a little bit of background of this presentation. So I will tell you a little bit about the tissue structure, about the background of the tissue, histology, barrier integrity, and other properties, about the enzyme expression. And then I will guide you through some of our or others' research using this tissue model and show you its utility in the assessment of drug safety, permeation of drugs and nutrients, drug-drug interactions, and other fields. So, what is this tissue model? Basically, MatTek has been long in the field of reconstructed tissue generation. This specific model is mimicking the structure and characteristics of the small intestine epithelium. It is generated from the normal healthy donor, so it's no cell line or anything. The healthy donor cells are collected from the healthy intestine. They are expanded a little bit in vitro on the petri dish. Then we freeze them in the huge lots. A lot of the cells are cryopreserved for a long time. And the aliquots of these cryopreserved cells are taken and seeded on the special inserts, tissue culture inserts, which are then cultivated on the air-liquid interface, which means that the nutrients are coming from the bottom, which is mimicking the uptake of the nutrients from the bloodstream, and the apical surface is exposed to air partially. There's a little bit of liquid on the top to mimic the intestinal content, and over time this tissue differentiates, and we end up with a model looking like this. This is the histology of two versions of the Epilntestinal model: the partial thickness, which has only the epithelial part, and the full thickness, which is also subepithelial fibroblast component. Basically, we believe that this is a very nice representation of the intestinal barrier or intestinal epithelium, as it has a proper composition of the cells, and it also mimics the morphology. The morphology of the small intestine epithelium, as you probably all know, is characterized by the presence of the villi. We have these villi-like structures. You can probably see better on this next slide. Here is the histology stained immunohistochemically of the full thickness tissue. So you can see the fibroblast layer. On top of it is the epithelial layer with these nice villi-like structures, which are covered by microvilli, as shown here by staining of the villi, which is the main component of these protrusions. You can see here that the tissue has intact tight junctions, even in the 3D morphology. And here on the bottom of the slide, you can see some electron microscope images showing the tight junction, which I mentioned before, the brush border. Here is one of the villi-like structures covered by the brush border, which is detailed here on the right. As we all know, the cell composition of the intestinal epithelium is consisting of several types of cells, besides the enterocytes, which is the main component, which plays a role in the absorption mostly of the nutrients and drugs and so on and so forth. There are some other cell types, such as Paneth cells, stem cells, LGR5 positive ones, Tuft cells, cells producing mucus, and Other types of cells, we have evidence for most of these cells being present. To be honest, the ratio of these compared to enterocytes is not exactly the same as in normal tissue, but they are present there, so their functions can be studied. Just to demonstrate a little bit on the longevity of the model, so the model can be kept in culture for over a long time, which is a big advantage when you want to do the chronic exposure to some compounds, drugs, or nutrients, or repeated exposure. You can see here that here is the tissue integrity measured using the epithelial electric resistance, which is measuring as I said, the integrity of the tissue or the tight junctions. You can see here over the period of 28 days, it only changes very slightly. In the main table, you just see the comparison of these parameters in many of the lots produced in 2 different sites of our company, in US and in Europe over here in Bratislava. You can see that these tier values are quite similar to each other, the averages, and the variation is quite low. There are many areas where the model has been used so far by us or by our collaborators and customers. Most of all, there is use in the study of drug toxicity, even more in the field of drug permeation, because it is very important to have a good model, physiologically relevant one, to really study the bioavailability and permeation and metabolism of the drugs when they are perorally used. But it has been also used to study the interaction with microbiota and with the pathogens. Wound healing is very interesting area, studying the compounds that might promote recovery of the damaged intestinal epithelium, inflammation, nutrient absorption, and so on and so forth. I will actually try to concentrate on these fields, the toxicity, permeation, and nutrient absorption, but there are some more areas to be explored. So first of all, a little bit on intestinal toxicity. A few years ago, we have done a study in collaboration with AstraZeneca pharma company. They approached us with a request to develop a model that would be able to predict the adverse effects of some drugs used in oncology. One of the common side effects or adverse effects is diarrhea, which is problematic because it causes discomfort, and it often results in treatment discontinuation and in modification actually of the absorbed dose of the given perorally used drugs. So we have decided to test our EpiIntestinal, at that time quite new model, and see if it can be used to predict such effects. We have obtained about 30 drugs from them with known properties in terms of adverse effects, namely diarrhea. We had these drugs on the left, which were causing diarrhea, and these drugs on the right, which were mostly safe or with only mild adverse effects. I'm not going into details, but we have tested several possibilities, and we ended up with a model where we used the previously mentioned method, measuring of the barrier integrity using transepithelial electric resistance. And we have developed a model where we treated the tissues over a period of several days with these drugs at 3 different concentrations, and then we looked at the point where the TER, the trans epithelial electroresistance, dropped by 15%, calling it TER IC15. And the ratio of this parameter, or the concentration at which the TER dropped, the ratio of this concentration with the maximum plasma concentration of the specific drug, gave us a parameter. And if this parameter was below 2, the drug from this group, all of the drugs for which this parameter was below 2 were causing diarrheas, while all the drugs which are not causing diarrhea had this parameter above 2, which was or about 80. This is logarithmic scale, which was pretty nice. We had pretty nice sensitivity and specificity. And to test the predictive value of this parameter, we then tested 4 drugs that previously failed in the clinical studies, but in the preclinical settings, they seemed to be okay. They didn't seem to be any adverse effects. So these drugs were tested on rats and dogs, or in vivo models, and as you can see, there were no or only very, very mild adverse effects in terms of the gastrointestinal tract, but in the initial clinical setting, they were causing quite diarrheas in quite a high proportion of the tested subjects. When we tested them on our model, interestingly, we have shown that all of them, the previously mentioned parameter was way below the threshold. So they would be all predicted as active as causing adverse effects, which suggests that this model might be quite promising for the clinical or preclinical use to test the toxicity of the drugs. Other area which was studied by our collaborators is the modeling of the disease. Here you can see one of the images from the study where our model was used to model the infection by the Shigella flexneri, which is an invasive bacterial pathogen. And the group from the Massachusetts University was using our model to study the mechanisms involved in this invasion. And here you can just see the images, the microscopy images showing that our model was successfully able to model the invasion into the epithelium, because this bacterium is able to invade inside the epithelial layer. You can see here transversal look inside one of the villi-like structures, and I'm not sure how well you can see it, but you should see the red stained dots of the bacteria invaded into the epithelium, while if the non-invasive strains of E. coli and Listeria monocytogenes were used, these are stained in green in this case, none of those non-invasive ones were able to permeate the tissue. As a spin-off of this experiment, we were also interested if our epithelial model is able to react to the presence of pathogens by the innate immunity reactions. The most epithelia contained receptors such as toll-like receptors and nod receptors, which are able to sense certain pathogen-associated molecular patterns such as endotoxin or liposaccharide, or muramyl dipeptide, or flagellin, and so on and so forth. So we treated our tissue by several of these pathogen-associated molecular patterns, or PAMPs, and we looked at the response, at the cytokine response. And as you can see on these few examples, the treated tissues tissues treated by these PAMPs produced all of the tested cytokines on these imagings. What was even more interested, if the tissue was treated by single PAMP, there was certain response, as you can see in the middle bars. But if the PAMPs were applied together, these two ones, muramyl dipeptide and lipopolysaccharide, the response was much more pronounced in terms of cytokine release, which has been described previously in vivo, and which was very interesting to see such a physiological response. On the other hand, it is known that prolonged exposure to some cytokines is also associated with some chronic conditions, such as chronic inflammatory conditions of the intestine, which is also associated by the disruption of the barrier of the intestine. So we were interested to see if the exposure to some of the cytokines which are known to be associated with the IBD, for example, inflammatory bowel disease, if it also has similar effects on our tissue. And you can see here two examples of the comparison of untreated tissue in blue after several days in culture, and the tissue which was treated by the IBD-associated cytokines. You can see that they actually really cause this disruption in vitro, which makes it a promising model to mimic such a disease. So far, we don't have a tissue that would be derived directly from the patient with the IBD or similar disease, but we can actually establish the condition in vitro. This is just to show you an example where the tissue was treated with these cytokines, and that this condition can be actually reverted. So this is the integrity in blue bars. This is the integrity of the control tissues. In red bars, you can see as the integrity drops over time after exposure to the cytokines that we have previously shown to cause the disruption of the integrity. And if the tissue was concomitantly treated with the anti-inflammatory drugs, you can see that the effect of the damaging cytokines was compensated. Another interesting area which we actually studied quite a bit recently in our lab or with our collaborators is the study of the wound healing or epithelial restitution. We can use the model to generate a wound. Here on this little video, you can see the progression of this wound. So basically, the one with the biggest hole is the one where the wound was generated, and over the course of the 6 days, approximately, you can see as the cells migrate from the border of the wound into the disrupted area and are able to recover fully the tissue. You can also monitor this using the tear. Here you can see the comparison of 3 different tissue in bluish colors. You can see the normal tissue with the tear maintained in roughly the same values or slightly increasing. This increase was, I showed you before that the tear is pretty stable, but here the increase, slight increase, was caused by the treatment, which was also used in this case. And in the reddish colors, you can see 3 tissues which were wounded. So this is the pre-wound state. Once we have wounded them, the tear dropped by about 40%, and over the course of 6 days, as the cells were growing into the area, the measurable integrity of the tissue recovered. You can see the same process also here in the histology. So now to the probably the most studied area, the nutrient permeation and metabolism. You can see here a study by one of our collaborators. They were looking at the usability of this model to study the drug permeation, and they were comparing it to the model which is well established in their lab, which is based on the cell line CACO2. This is also used by many other labs. But as you can see, when they compared our model to CACO2 and they compared both results to the actual in vivo results from the human absorption of these drugs, I'm not going into details which were they, but you can see much better correlation with normal absorption in case of EpiIntestinal model than in cell line based model. This is just the summary of this experiment. So actually I have the details here showing that they can be split into two groups, high and low. And if we use this split, we can calculate the prediction model, and we can see that EpiIntestinal can achieve pretty nice sensitivity and specificity in predicting drug permeation. There are some more studies done by our collaborator. This study, which I will mention here, by one of our customers, also has some interesting data on the permeation, but I will pick a little bit different area. They were also looking at drug metabolism, because as you probably know, there is quite a important portion of the metabolism of drugs going on even during the permeation through the intestinal barrier via different kind of enzymes such as cytochrome P450 and its isoforms. You can see here comparison of EpiIntestinal and CACO2 based model. They were looking at midazolam drug, and you can see that in CACO2 they were able to recover after the permeation almost 100% of the drug, while if you look at this bottom line, there was only about 50% recovery in our tissue, which was not very nice. It would suggest that the permeation of our model is lower than in terms of CACO. On the other hand, we know that midazolam should be metabolized into hydroxymidazolam. So the theory was that this undetected part of the midazolam was actually the conversion product, and this was confirmed when the enzyme responsible for conversion was blocked. The recovery in our model was much higher, while this didn't change in CACO2 based model, which suggests that the metabolism doesn't work properly in this model. This was also confirmed by the reverted experiment when they were looking at the hydroxymidazolam, which is the metabolite, and there was a nice recovery of this metabolite in our model, but not in CACO2. This is just to show you that besides the CYP3A4, which was enzyme which was utilized in previous slide, there are other enzymes which are important for metabolism of drugs and other substances are present and expressed in our model. A little bit to nutrients finally. We were studying this model in dynamic condition with the collaborator from the React for Life company, the group of Silvia Scaglione. We were using their vivo device to cultivate the tissue in the dynamic conditions, which even increases the physiological relevance of the model by providing the simulated stream of the blood underneath the tissue. And you can see that here is just an excerpt of the published paper. We were looking at two different sugars. One of them is lactulose, which is used in vivo to monitor the integrity of the tissue, and which should not be absorbed by the intestine. And you can see here that it's actually through the EpiIntestinal properly mimicked the non-permeability for the lactulose, as the recovery was below 1%, and it was not changing with changing concentration of the lactulose, while the permeability of mannitol, which actually is permeable, was much higher, and it was concentration dependent. This is also very interesting because the previously mentioned CACO2 model is virtually impermeable for mannitol. The mannitol is actually used as a control of integrity in this kind of models, but it's not really physiological because mannitol is absorbed in human. These are the figures I borrowed from this paper from one of our customer. They are developing different kinds of encapsulations for micronutrients. These types of encapsulations are intended to protect these nutrients, such as vitamins or iron, from the physical conditions associated with the storage or cooking. Here is just their process as they encapsulate the vitamin, for example, with hyaluronic acid, and the polymer, which is based on butyl metacrylate, I think, some sort of, not a specialist here. Unfortunately, this encapsulation initially was shown to decrease the bioavailability of the nutrients. Here is the study of on the volunteers. You can see here that free iron was absorbed at certain level, but if the coating, the encapsulation was involved, the bioavailability decreased quite considerably. So they used our model to look if the presence of these encapsulation agents, the hyaluronic acid and butyl metacrylate, affects the bioavailability. And actually, they have shown that while hyaluronic acid didn't change the absorption too much, the percentage of the butyl metacrylate was actually very important, as when they used the original ratio of iron and the BMC, the bioavailability was at about 30%, while if they were decreasing the ratio of the BMC, the bioavailability increased quite considerably. And when they modified actually their final product and tested on volunteers, they have seen quite an improvement with the bioavailability of about 89% of the free iron, while they were able to protect these nutrients from physical conditions associated with storage. Last little study, which is sort of ongoing, we are studying the effects of the fruit-based enzymes, in this case, papain and bromelain. These are hypothesized to be used to improve the absorption of non-protein drugs, because they are known to affect the paracellular transport of some nutrients by slightly modifying the integrity of the intestine. And using we applied these enzymes on our model, and we're studying the effect of these on the integrity. As you can see here, for example, bromelain, you can see that after 30-minute exposure, it dropped in the integrity as measured by TEER, and it was also associated with quite considerably increased uptake of the lucifer yellow, which is impermeable fluorescent dye, in normal condition impermeable, but after the treatment with bromelain and also with papain, its uptake increased quite considerably. And you can see here the uptake also on the fluorescent images from the histologies. So this is it, what I meant to present today. Just to summarize a little bit, we have this tissue from the normal donor, which mimics the structure and function of human intestine. It can be cultured for considerable time, which allows the different modalities to study the uptake of nutrients or effects of different compounds on the tissue after the peroral dosage. It can be exposed in acute and chronic way this way, and also it can be exposed by different modalities because it's a planar thing, so you can mimic the sort of luminal exposure by applying the compounds on top of the tissue, or the systemic one coming from the bloodstream if you add your compounds into the basolateral medium. And as I have shown you, it can be used in different areas of your research. These are just few of my collaborators and the people who were actually involved in the development of this model, which is developed by the group led by Dr. Ayehuni, which is my colleague from the US lab. And I would like to thank you for your attention, and I'm looking forward to your questions and comments. Thank you very much.